All right, good morning, afternoon, whenever it is that you're watching. I just wanted to put another video here for you guys to take a look at. The topic of this video here today is natural selection, genetic drift, and gene flow, which are all factors that can cause allele frequencies to change. So, what we want to take a look at today is how do natural selection, genetic drift, and gene flow cause allele frequencies to change, and also give a little bit of an explanation of what are natural selection, genetic drift, and gene flow. What do those things mean when we talk about those? So that's what we're going to get into today. I'm currently on day 5 of the quarantine beard, also here as I'm making this video. So it's starting to come in a little bit more. All right. So the first thing I want to talk about with you is the hugely important concept of natural selection. It's so important that of the 8 units that the College Board has given us to teach this year, Unit 7 was called natural selection. So, when we talk about natural selection, what that means is differential success in survival and reproduction here. Okay. And reproduction. That is hugely important right there. Okay, so um, as we take a look here, this don't forget it's not just survival of the fittest; it is survival and reproduction of the fittest. Okay, and that is due to variation in heritable traits. So, what does that mean? Uh, that means that because we inherit traits that are different from other individuals, because we all have a little bit of variation between one another, and all organisms have some degree of variation. Certain variations will give organisms an advantage over others that within their population, so they survive better. And they reproduce more often. Okay, that's what we mean by differential success. Some individuals survive better and live longer, and also reproduce more often and leave more offspring than others. That's what differential success means. What happens because certain individuals survive better and reproduce more often is that the alleles in one generation are passed to the next generation in a different proportion than they exist in the current generation. If we take a look over here at this little diagram, this shows you kind of what I'm talking about. Like in the previous video, we mentioned that mutation can generate variation within a population. So we had this initial population where everybody was the same, some mutations occurred, and we get some variation in this population. Now, the super light individuals were selected against. The darker individuals survived, passed down their darker alleles, and again, the lighter individuals were selected against. And so now the population looks a lot different than it did initially, and certain alleles are not being passed down with the same frequency as others. The darker alleles are being passed down at a higher frequency than those lighter alleles were. So it's important to know that natural selection is the major driving force behind evolution, but it is not the only factor that can cause organisms to change over time, right? So if we take a look at the next slide here, um, this is just an example that I gave you. This is not necessarily a real life example, but again, this shows you, okay, here we have a population of lions with variation in coat color, right? The lighter colored lions are able to blend in better and sneak up on their prey, and capture prey, so they survive better. They live longer, they reproduce more often. So what we see in the next succeeding generations is that the lions have a lighter coat color in general in the population. Those dark coat color alleles really aren't getting passed down very often. So we see the allele frequency for coat color has changed, where initially there was an equal amount of light and dark coat color alleles, but now we take a look and they're almost all light colored alleles. Okay, so that is an example of natural selection. Another example that I can give you came from Drosophila melanogaster. who those are our fruit flies, and they have an allele for insecticide resistance, including DDT, which is a really strong insecticide. In any lab populations that were established before the 1930s, say like in Thomas Hunt Morgan's labs, the frequency of that allele for insecticide resistance was 0% if those populations were established before the 1930s. However, in any populations established after 1960, we see that the frequency of that allele is 37% because that DDT and insecticides started to get used in nature between the 1930s and 1960s, and that then is a very strong selective pressure for the Drosophila that had happened to have that allele for insecticide resistance. They survived more often and lived longer and reproduced more often, so they passed down that allele for insecticide resistance. All right, so next topic that can affect allele frequencies in a population and can cause allele frequencies to change, so can drive evolution, is something called genetic drift. Now, I know this picture is a little bit different than the one that you have, but I figured two different visuals of genetic drift would not be a terrible thing. But genetic drift, when biologists use that term, they're referring to chance events that alter allele frequencies, right? So here we have a population of beetles. Some are green, some are brown. And even though maybe these green beetles are better adapted to their environment and normally would survive better and reproduce more often, if somebody happens to walk past and just accidentally step on the green ones and miss the brown ones, well, now the allele frequency for the brown color in these beetles has drastically increased just due to a random chance event. And in the next generation, most of the beetles will inherit that brown allele, and there will be very few green beetles in the next generation. It had nothing to do with one organism being better suited or adapted for the environment. It just happened to do with a random chance event. Okay. Now, one thing to know about genetic drift, it is more likely to alter frequencies, gene frequencies in small populations. For example, if you were to flip a coin 10 times and it came up 7 heads and 3 tails, would you be very surprised? Probably not, right? That's a pretty small sample size. It happens. But if you flipped it 1,000 times and it came up 700 heads and 300 tails, now you would start to think there's something going on with this coin, that it's not a fair coin, right? So again, if random chance events are more likely to occur and change things in small populations than in large populations. One example of genetic drift of a random chance event can be seen when we have what's called the founder effect, when a few individuals become isolated from a larger population. One example of that would be in 1814, there were 15 British colonists that settled the island of Tristan da Cunha. And one of those 15 individuals carried an allele for a progressive blindness disease called retinitis pigmentosa. And of the 240 descendants living on that island in the 1960s, 4 of them had this recessive trait of retinitis pigmentosa. The allele frequency for that particular allele in the population is 10 times higher than in the population where those founders came from. You can see in this picture here, we have a few individuals here, and they moved to a couple of them moved to another island, and now the yellow allele is much higher in frequency in this new population than it was in this old population. And we see that Native Americans have a really high frequency of blood type O compared to their ancestors where they came from, because again, it was just a small group of individuals who founded that Native American population. Another random chance event that can cause allele frequencies to change is what's called the bottleneck effect, which is a drastic change in allele frequencies due to a sudden change in population size. So the population goes through a bottleneck in size, like you see here. Right, here's your original population, only a few individuals survive some event, and your surviving population has an allele frequency that is much different than your original population. Originally, we had about equal numbers of blue and white alleles with a few yellow ones thrown in. Went through a bottleneck. Now we have a lot of blue and just one white and no yellow left in this example. So this population may increase in size again, but it would have very little diversity after this after this bottleneck effect, because these individuals, this small population, has to interbreed with one another to found the new population. That's what happened to cheetahs. That's what you can see happening here in this graph. Population size undergoes a drastic reduction. Genetic variation does too. Now you have just a very few individuals to found the population, and the population may grow again, but there are very few genes to choose from here. And so the genetic variation really isn't going to change very much as this population grows in size. You're gonna have a lot of closely related individuals. And that's what we see with cheetahs, doing a genetic analysis of cheetahs who went through a massive bottleneck during the Ice Age. Population, they almost went extinct. And now the genetic diversity of cheetahs is very, very, very small. All right, now, genetic drift is significant in smaller populations. It can cause allele frequencies to change at random. So it's just a little review here. It can cause a loss of genetic variation within a population, and it can cause harmful alleles to become fixed. In other words, perhaps the only individuals that survive actually have a harmful recessive trait, and that becomes fixed in that population. That harmful recessive trait will just continue to be passed down and passed down and passed down. Okay. All right. Another factor that can affect genetic diversity within a population, it can cause allele frequencies to change, is what's called gene flow, which is the movement of genes into or out of a population. So when new individuals move in or old individuals move out, then we have gene flow, right? So when we have migration, either immigration or emigration, then we have gene flow. And what actually ends up happening is neighboring populations can actually become more genetically similar to one another. So what you see here is a specific type of grass called bent grass growing on top of a copper mine. Now copper, that copper would get into the soil that this grass is growing on, and copper is toxic to most types of grass, and it's toxic to bent grass. But there are some individuals who actually have alleles that allow them to tolerate the copper that is in the soil. And so those alleles spread quickly through the population as the grass reproduces. Now, over here in the background, you can see an area where this is not on top of a copper mine. And so the thing about that is that on soil without copper contamination, the copper tolerant plants actually don't grow as well as the ones who are not copper tolerant. So you would expect you would not find any copper tolerance alleles in the grass over here, and you would find only copper tolerance alleles in the grass growing here. That's not the case, however, because grass spreads its pollen and reproduces through by pollen being spread by the wind. So the pollen that this grass over here is producing is going to blow into this population. The pollen that the grass over here is going to blow into this population. And so these two populations are actually more genetically similar than you would predict necessarily based on the environments where they are growing. Okay. Gene flow does introduce new genes into a population at a higher rate than mutation does, because it happens much more often. And once the new alleles are present in a particular population, then natural selection will act on them. Okay. All right. So one term that is commonly used by biologists is what's called relative fitness. And what this does is it's a way to measure the genetic contribution an individual makes to the next generation relative to others. So if we had a hypothetical wildflower population where some of the flowers had red alleles and others had white, if the red flowers produce more offspring than the white flowers, we would set their relative fitness at 1 and then compare the white flowers to that. So if the white flowers produce only 80% as many offspring as the red flowers, their relative fitness is considered to be 0.8, right? So we're comparing how fit the two different flowers are to one another. That's why it's relative fitness, right? And fitness is a measure of how much of what kind of a genetic contribution an individual makes to the next generation. I love this one. Okay, so if an individual is sterile, then its relative fitness is zero, no matter how much longer it lives than others. So the fittest organisms are not the ones that live the longest. They are the ones that have the greatest genetic contribution to the next generation. So if an organism is sterile, then it has a fitness of zero. Um, so organisms must survive to reproduce, obviously. And it's important to remember that natural selection acts on phenotypes. And by doing that, it's then indirectly acting on genotype and changing allele frequencies, right? But in our lion example, natural selection was working on the yellow color versus the darker brownish color, right? So it was working on the phenotype, but then the alleles that create that phenotype are passed down to the next generation. All right, when we take a look at what happens to populations as natural selection acts, we actually see three different patterns emerge within populations. The first one is directional selection, where one extreme phenotype is favored over another. Here we see an initial mouse population has a nice bell curve of frequencies of different fur color, but perhaps these black mice blend in with the background and hide from predators better than the white ones. Maybe they stand out quite a bit. And so we see that over time, the one extreme phenotype was favored and then the other was disfavored. So we see a shift in frequency to the darker color being favored. Okay. When we have what's called disruptive selection, that is when the average phenotype is selected against, and both extremes are favored. So maybe these mice here live in an area where some parts of the environment are really dark and other parts are really light. So these guys can live in the darker areas, these guys can live in the lighter areas, and if you're in the middle, you stand out whether you're in a dark background environment or a light background environment. And then finally, we have what's called stabilizing selection, where the average phenotype is favored. So maybe both the black and white mice stood out here. An example in humans of stabilizing selection is average birth weight of babies. Extremely large babies were selected against prior to the development of cesarean section, because both the mother and child sadly would die during childbirth. And then very small babies obviously had a very hard time surviving. Their body systems might not have been fully developed yet. Their immune system might not be fully functioning yet. So small babies are also selected against. All right. So one thing to remember, all these different factors, natural selection, genetic drift, and gene flow, all change allele frequencies. But natural selection is the only one that is consistently working all the time. Random chance events don't always happen within a population and aren't always taking place. So genetic drift is not always occurring, and individuals are not always moving into or out of a particular population. Okay, so one interesting aspect of natural selection, or maybe type of natural selection here, is what's referred to as sexual selection, right? So this is something that factors into natural selection because individuals with certain traits are more likely to obtain mates and pass down their alleles than others. And this leads to what's called sexual dimorphism, where males and females of the same species look very different from one another. Especially if you think about birds, for example, we see that. So in intrasexual selection, members of the same sex compete for mates. That's usually between males, and that leads to some different behaviors with those males. So let's take a look here at this video. Make sure the volume's up. See, we have members of the same sex competing with one another. Okay, so that was one example of intrasexual selection that selects for particular behaviors in some organisms, particular appearances in others. Let me put the wrong one. I guess I don't need the anatomy one here. Um, so um, Again, that was intrasexual selection, where there's competition between individuals of the same species, leading to the development of particular behaviors, sometimes particular appearances. But the other type of sexual selection that we see is what's called intersexual selection. And it's usually females choosing a mate. Females tend to be very choosy in selecting a mate of the opposite sex, and that leads to male showiness, right? Because the showier a male is, the louder he is, that tends to indicate the healthier he is and the more fit he is. So here we'll see an example in this one of intersexual selection leading to the development of male showiness and interesting behaviors. All right, thanks, Neil. She spent a lot of time by herself in the forest. No, it's not what you think. This is my favorite video in all of biology coming up. All right. So um, I really enjoy that last part right there with the moonwalking birds. Just trying to get this back down. There we go. But again, that showed you some male showiness that has evolved over time as females choose showier and showier traits. So um, we know genetic variation is super important in organisms being able to change over time and becoming better adapted to their environment. How is that genetic variation preserved? Well, most organisms are going to be diploid, so a lot of genetic variation is hidden from selection by heterozygotes in what's called heterozygote protection. That those heterozygote individuals are actually protecting those recessive alleles, and that maintains a large pool of alleles in the population. We don't get fixed at a particular locus, right? This was at a roadside in Minnesota back in 2002. I made my family pull over so I could take a picture in front of that sign. I mean, homozygous, how often you see that out there? Okay, so balancing selection refers to natural selection that maintains two morphs or phenotypes in a population, and that helps to preserve genetic variation. This happens through what's called heterozygote advantage, and then frequency dependent selection. Heterozygote advantage is what occurs when heterozygotes have a survival and reproductive advantage over either homozygote, like we learned about with sickle cell anemia, where heterozygotes had protection from malaria, but also did not have the full blown sickle cell disease. So I'm not going to go back into that in detail. We've done plenty with it. Frequency dependent selection, though, is one that maybe you're not quite as familiar with. This is what we refer to when we're talking about a particular phenotype's fitness decreases if it becomes too abundant. So in Lake Tanganyika in Africa, there are left and right-mouthed fish in that lake. What right-mouthed means is that their mouths actually curve to the right. And left-mouthed fish, their mouths curve to the left. And so when those right-mouthed fish, the way that they capture their prey is that they come up behind them and they need to sneak up on the left side so that their mouth that's curved to the right can then attack. And the left-mouthed fish sneak up on the right side of the fish that they of their prey, and their mouths curve to the left. So as they sneak up on the right side, their mouths are in position to attack. So what happens though is if the right-mouthed fish becomes more abundant in one particular year, then the prey fish learn to be more wary of what's coming up from the left side. And so the right-mouthed fish then are less successful at obtaining their prey. And the left-mouthed fish then survive better because they're better, they do a better job of getting their prey, and they become more abundant in the next generation. Then in the next generation, the prey fish become wary of a predator coming up from behind them on the right, and so the left-mouthed fish do not survive as well, and the right-mouthed do. So as a particular phenotype increases, it's the fitness of that phenotype goes down in this particular example. Okay. And then one other way that genetic variation is preserved and differences in individuals are preserved are in neutral variations, where we have nucleotide differences between individuals, but they give no selective advantage because they occur either in non-coding DNA or they are silent mutations. And so the only thing that would affect the frequency of those particular genetic variations would be a random chance because nature wouldn't select against them. They don't show up in the phenotype. Now, one argument that we get a lot is why doesn't natural selection fashion perfect organisms, right? Um, well, for one thing, natural selection can only act on existing variations. Advantageous alleles don't arise just on demand, right? Like I can't just be like, oh, I wish I had wings and just grow some wings because I developed some alleles that allow me to grow wings, right? Natural selection can only act on what's already present, right? And so that's kind of what I'm showing here, right? It'd be a huge advantage if this were able to happen for these beavers, but it just can't. Evolution is also limited by historical constraints. It can only take existing structures and modify them. It does not create new ones just from complete scratch. For example, the wings of birds are actually adapted from the forelimbs of their ancestors with feathers on them and maybe some webbing. But they are just adapted from the forelimbs of their vertebrate ancestors. They weren't just grown from scratch. Adaptations are often compromises. If you think about it, some organisms live in a couple different areas, right? Like seals, for example. They walk on rocks, but they also have to swim. So they could walk on the rocks better if they had fully formed legs with feet on them, but then they wouldn't swim nearly as well with if they had those. Or they could swim a lot better if they had fully formed flippers, but then they wouldn't be able to walk on land as well. So adaptations often have to be compromises based on an organism's way of surviving. And then we got to keep in mind that chance and natural selection and the environment often interact. Like genetic drift, random chance events could eliminate some alleles from a particular population, and then natural selection can only act on what's left. For example, if some birds or insects are blown hundreds of kilometers over the ocean to an island, not necessarily those from the original population that would have been best suited for that island ended up there. They just happened to get blown there by a random chance event, some massive storm or something. And so then natural selection can only act on what is available, right? And so that's why we don't get these perfect organisms. A few of these natural selection things. Okay, so that's it. That was kind of a longer one. But there's some really important stuff there, especially in natural selection. So hopefully you guys are staying safe, staying healthy, and I will check in with you guys in a little bit.